Hereby I open this academic ceremony in which Amelie Heesch will defend the academic thesis, Endogenous radiotherapy of triple-negative breast cancer, the prostate-specific membrane antigen as a vascular target. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Prorector, dear members of the Defense Committee, dear family, friends and colleagues on site and online, in the next 15 minutes I want to introduce you to the topic of my PhD thesis, which is called Endogenous radiotherapy of triple-negative breast cancer, the prostate-specific membrane antigen as a vascular target. The first step in the diagnosis of breast cancer is actually the detection of the tumor, which is done usually via clinical examination of the breast and also via imaging, for example, with mammography and ultrasound. After that, a biopsy of the tumor tissue is taken to analyze the tumor grade and also the receptor status of the cells, which is needed to classify the respective subtype of the breast cancer. Afterwards, a tumor staging is done. Here it is checked what the actual tumor size is of the primary tumor and if there are any metastases in the body of the patient. In some cases, there are also genetic tests necessary, and if all these steps are done, then the therapy for the patient is planned. And here it is very especially necessary to know the receptor status of the cancer cell to classify the subtype, and after the classification, then the treatment can start. So here you can see a tumor of the breast, and if we take a closer look on the tumor cell itself, we can see that on this breast cancer cell, 3 different receptors are expressed, which are called progesterone receptor, estrogen receptor, and HER2 receptor. And these receptors serve as drug target, and for these receptors, we already have drugs which can be used for the treatment. These ligands bind specifically to the receptor and then are used for the targeted therapies. So depending on which receptors are expressed on the cell surface, the respective subtype is classified. So we have, for example, the luminal A breast cancer, which expresses the ER receptor and the PR receptor. We have the luminal B breast cancer, which expresses the ER receptor and the HER2 receptor. Then we have the HER2-enriched subtype, which overexpresses only the HER2 receptor. And the last subtype is the TNBC subtype, so the triple-negative breast cancer. And as you can see, it does not express any of these 3 receptors, which is also one of the reasons why it is harder to treat than the other subtypes and also has a worse prognosis. So the question now is, if we don't have a therapeutic target on the cell surface itself, like maybe there's another way to find any other therapeutic targets. So we could maybe check the surrounding of the tumor. This is called the tumor microenvironment, and this tumor microenvironment consists not only of tumor cells itself, but of several other subtypes, several other cell types. For example, these cancer-associated fibroblasts you can see here, or the tumor-associated macrophages, and also the tumor-associated endothelial cells, which line the blood vessels of the tumor vasculature. So these 3 groups I already told you are at the moment in the main focus of developing new therapeutic targets for breast cancer. And for my project, of course, because I analyzed the potential of PSMA as a vascular target, we will take a closer look at the endothelial cells and the blood vessels of the tumor. So the tumor-associated vasculature is very important because it provides the tumor with nutrients and oxygen, and is therefore very essential for tumor growth and metastasis. And interestingly, it was found actually that this tumor-associated vasculature expresses PSMA on the blood vessels, and this is also the case in TNBC. So luckily, there's already a drug which binds specifically to the PSMA receptor. This is called 177 lutetium PSMA, and this is at the moment used in the treatment of prostate cancer patients. So through this ligand, which will then bind specifically to the PSMA receptor, the radiation would damage the tumor vessels, and therefore the tumor growth would be inhibited. So we asked ourselves, is PSMA actually a potential target for TNBC? And therefore my project was divided in two parts. The first part was the in vitro study, where I did a lot of cell experiments and cell analysis. And in the second part, I did a preclinical study where I used immunodeficient mice, which were xenografted with cancer cells. And also here, of course, I had a few questions which I wanted to answer. The first one, is actually PSMA present on the tumor vessels? Because, of course, for the PSMA targeted treatment, we would need a target on the blood vessels. Is the drug specific? Then, of course, also if the therapy is efficient. And also important, if you are trying to develop a therapy, if the therapy is safe and tolerable, and if there are any side effects. So I will start with the in vitro study. Here you can see a so-called trans-well system. So this is like a co-culture of cancer cells with endothelial cells. And yeah, as you may remember, these endothelial cells are the cells that line the blood vessels. So we somehow mimic the tumor vasculature in vitro. In the top chamber, I cultivated the cancer cells, and in the bottom chamber, I cultivated the endothelial cells. And they are only separated through a very thin membrane, and this membrane is only permeable for very small molecules, but not for the cells itself. So I let them grow together for 4 days. So the tumor cells start, over time, they start to secrete several factors, and they start to secrete several factors, which then influence the endothelial cells and their behavior. And the endothelial cells, as a response, start then to express PSMA on the surface. After that, I discarded the cancer cells and only analyzed the tumor cell, the endothelial cells, and checked, for example, for PSMA expression. I also had, of course, a control situation because normal endothelial cells that are not tumor-associated should not express PSMA. Therefore, I did not co-cultivate any cancer cells and only co-cultivated the endothelial cells alone, so there will be no factor secreted, and therefore the endothelial cells will not start to express PSMA. Here you can see an antibody staining where I stained these co-cultivated endothelial cells, which were co-cultivated with cancer cells. In green, you can see the PSMA signal. In red, you can see the CD31 marker, which is an endothelial cell marker, and which appears yellow. There we have the overlay of the green and the red signal. So as you can see here, after the cultivation with the cancer cells, the endothelial cell actually start to express PSMA. Yes, and this was not the case in the control situation where we only cultivate the endothelial cells. We can see that it only expresses the endothelial cell marker, but no PSMA. I also quantified this because I wanted to know how much PSMA actually is induced. So I analyzed a huge panel of cell lines and checked for the PSMA expression in the endothelial cells, which they were co-cultivated with. And I found that actually the highest amount of PSMA induction was in the was done by the MDA MB231 and the BT20 cells. So they had the highest amount of PSMA induction actually in the endothelial cells. So I picked one of these two very high inducing PSMAs also for the xenograft in the preclinical study. So here we injected tumor cells, which in this case were the MDA MB231 cells, and they were injected, and after the tumor reached a specific size, then we started the 177 lutetium PSMA therapy. The radiotracer was here injected into the tail vein of the mice. I used in this preclinical study 3 animal groups. So the first one was the control group. These mice did not receive any radioactivity. They only received sodium chloride as a control. Then I had the group which received the single dose of 177 lutetium PSMA. The total activity here was 60 MBq. And I also had one group which received a fractionated dose of lutetium PSMA, and these mice received 4 times 15 MBq, which was injected every 7 days. So to show you what the outcome actually of this preclinical study was, we could actually see that the treated mice had a significantly smaller tumor volume compared to the control mice. So also here the mice which received the single dose had even a smaller tumor volume compared to the mice which received the fractionated dose. And this also reflected in the survival of the mice, where I could see that the mice which received the single dose of 177 lutetium PSMA lived up to 42 days. The mice which received the fractionated dose lived up to 37 days, and the mice in the control group only lived up to 30 days. So I actually found that PSMA is a potential target for TNBC, and I could prove that PSMA is actually present on the tumor vessels. I also saw that the drug binds specifically to the tumor-associated vasculature. I also could see that the therapy has an effect, that it's efficient, and also here the single dose was a little bit more efficient compared to the fractionated dose. And I also checked for side effects, so I analyzed the organs of the mice and could not see that they took any damage, and also the mice itself showed no abnormal behavior or anything, or weight loss. Yes, and with that I want to thank you for your attention and thank everyone who supported me during my PhD journey, and now I give back the word to the prorector.
Thank you for your presentation. The opposition will be opened by Professor Zuhausen, Professor Pathology at Maastricht University. Professor Zuhausen was also the chair of the assessment committee.
Thank you very much. Congratulations, dear candidate. Congratulations with your thesis, with your book. And I would also like to extend my congratulations to your team of promoters and to your supervisors. And let's start this defense by letting others do the work. May I ask one of your paranymph, one of your colleagues, to read out, I think, statement number 10.
Number 10. Research is to see what everybody else has seen and to think what nobody else has thought.
So what is it, what everyone has seen and you're the only one who thought?
Yeah, I really like this quotation actually because, I mean, the quotation itself shows a little bit like what the spirit of research is. So at first you start research with observations and you gather information and get results. And the real challenge after this is to take this in a higher context and in a bigger picture. So actually it's not enough to only observe things, but you have to make a new approach out of it. And in my case, for example, in my project we had the thing that we already knew that the PSMA is somehow on the vasculature, and we already had this radioligand therapy, which is used in prostate cancer, but this new approach to use it in combination with TNBC is actually what nobody else has thought. So
very fine answer, thank you very much. May I take you, because this is a great biochemist, Hungarian biochemist, may I take you to page 57 of your chapter 3 and figure 1. And I'd like to discuss some of your figures. And as a hobby biochemist or hobby researcher, I very much like this. Actually, one of your co-authors introduced me 20 years ago to alternative splicing and Maastricht, and we had many papers on alternative splicing, so I really appreciate this paper.
Yes.
So in figure 1, you state actually that by using RT-PCR, that the full-length PSMA transcript was expressed in all TNB cell lines except HCC 1937.
yes.
And why do you think I do not agree with this when I look to figure 1? And I'd like to take a closer look at lane 6 to lane 8. Something is different there, isn't it?
I mean, you are talking now about the PCR results,
Yeah.
right?
Yeah, figure 1. And the upper row is the upper panel, PSMA, full length, 424 base pairs. And in my opinion, it's the lane 6 to 9, what is it, 6, 7, 8?
Mm-hmm.
Yeah, they are on different sizes, or aren't they?
Yes, I mean, it could possibly be that this is somehow like a modified version of PSMA, that it's like a little bit longer. That could be, but I think the thing is that for HCC 1937, we could not see a band, so.
Yeah, you conclude that this is in fact full-length PSMA. It seems to be even longer because it's a little bit higher in these
Yes.
3 cases. Did you sequence the PCR products?
I'm sorry?
Did you sequence the PCR products? Did you?
Sorry, I did not understand it
you can
acoustically.
excise or you can take the PCR product of your
Yes.
RT-PCR, and you can sequence them. Did you do this?
Yes, no, we did not sequence them.
Okay.
Actually, we only checked it in the PCR, and then the amount of the endothelial cell expressed. Then we did a qPCR with that, but not for the normal constructs.
Okay.
And
And
no
did you have a look in RNA sequencing results of these cell lines, or?
No, I mean, this is actually like an mRNA actually, which we transcribed into cDNA, but no, we also did no RNA sequencing.
The next thing is, of course, I'm a pathologist, and so I'd like to promote our pathology. The question is, did you have a look, or what is the difference between those tumors? Did you have did you look up the original reports from which these cell lines were made? Because they seem to be distinct from the rest. And so the question is, do they somehow differ in their pathology from the rest of the TNBCs which you were using? Because we all know it's a wastebasket diagnosis for a TNBC. So,
I mean, in general, these cell lines all somehow differ from each other because they carry different mutations. So, for example, we have some cell lines which exhibit the BRCA mutation. We also have some cell lines which which have a BRCA wild type. We have cell lines which derive from the primary tumor, and we also have some which derives from metastatic lesions. So, yeah, but you mean especially the HCC and the HS578, right?
yeah, but this HCC 37 is for sure negative. And basically, I think the time has already progressed, so I will return the word back to the prorector, and we'll be happy to answer questions if time is left over later. Thank you very much for your answers.
Thank you. The opposition will be continued by Professor Gonfin, Professor Nuclear Medicine and Molecular Imaging at the University of Leuven. Professor Gonfin was also member of the assessment committee, and we highly appreciate your participation to this academic ceremony.
Dear Mrs. Candidate, first of all, I want to apologize for not being present with you in Maastricht to attend your PhD defense, and I'm very happy that modern technology allows me to attend online. Second, I want also to congratulate you on the work that you did during the PhD trajectory and also for providing a clear PhD thesis, which was nice and interesting to read. So I also prepared some additional questions for you. First question, when we look at the activities of the lutetium PSMA INT that were administered in the mouse model, we can see that these are significantly higher relative to the weight of the mice than what we typically use in humans. While the efficacy was demonstrated with a reduction in tumor growth and increased survival, it was inhibited, but it was not completely stopped. When you think about potential challenges and considerations regarding translating these results into a clinical setting with typically a lower relative dose, which might be these challenges, and how might a planned clinical trial address these?
I think there are still a few challenges, of course, in the translation to the clinic, because of course, the mice differs as an organism from the human, and the mice have a very much higher metabolism actually than humans. Also, of course, this mice model had some limitations. For example, that the tumor heterogeneity was probably not quite comparable to a human, so maybe it would be an idea to do this study with a patient-derived xenograft, so to imitate better the tumor in humans, actually. And yeah, we also had a little bit problems with the necrosis, which was forming inside of the xenograft. So this was also a little bit difficult because due to this necrosis in the core, we had less vital tissue actually to treat actually. So yes.
Yeah, there was also a second question that I had regarding the necrosis and the suboptimal vascularization in the model. So what do you think were potential effects on the observed efficacy of, on the one hand, the relatively high activity? Would you expect that also in patients with lower activities this would be sufficient? And second, what are the potential observed effects on of the necrosis and the suboptimal vascularization?
Sorry, could you maybe repeat the first question?
So the first question was whether you how do you think that the limitations of the mouse model, given the necrosis and maybe suboptimal vascularization, how this affected the observed efficacy of the lutetium PSMA?
Yes, highly esteemed opponent, thank you for your question. I actually think that the necrosis somehow affects the whole treatment itself, because on the one hand we have, of course, less angiogenesis in the tumor. Therefore, we also have less therapeutic target, and therefore it could be possible that the tumor growth in the control mice would be even higher compared to the control group if we had from the beginning more vital tissue, so the tumor growth could be possibly even higher in the control mice with no necrosis in the core actually.
Okay, and then your promising preclinical results have led to the initiation of a clinical trial, or to the extent possible based on the available information. Can you explain some of the key aspects of the design?
You mean of the clinical study?
Yes.
Actually, I'm sorry, but I don't know any details yet about the clinical study. I'm sorry.
So what would be your advice then, based on the studies that you did? What would be your advice? What should be taken into account? Are there any specificities regarding patient selection, PSMA expression, and so on, dosing, activities? What would be your advice for the trial design?
So my advice would be, on the one hand, of course, that the patients should have proven PSMA expression actually in the tumor. That would be the first thing. Then I would say that what else could be criteria? For the amount of activity, as from what I saw, I would try a single dose approach. Of course, you could combine it with a fractionated in another group, so it depends on how many groups you will design for the study. And yeah, as the 177 lutetium PSMA is also in prostate cancer patients pretty tolerable, so I think there will be not a lot of side effects expected, actually.
Okay, thank you. Then one final short question. Would you see any other tumor types where, based on your current research, it may be interesting to start with lutetium PSMA treatment, non-prostate cancer?
I mean, there are actually a few candidates which could be a potential or could be potentially used with PSMA targeted therapy, because there are a lot of solid tumors which actually express also PSMA in the neovasculature. This could be, for example, renal cell cancer. It could be a bladder cancer, for example, which could have potential for this therapy. I know that glioblastoma also expresses a lot, but there I think we would get a problem with the blood-brain barrier, because the PSMA ligands are probably too large to reach the brain. So yeah, these would be a few candidates actually, which could be interesting, yeah.
Okay. Thank you very much. I give the word back to the pre-rector. Thank you.
Thank you. The opposition will be continued by Professor Joning, Professor Pathology at Aachen University. Professor Joning was also member of the assessment committee.
Thank you very much. Mrs. Candidate, I would also like to express my admiration and congratulate you on the diligence of your work, on the presentation in your thesis, and of course on the introduction and the presentation you've given so far. I think Professor Zuhausen just referred to triple-negative breast cancer as sort of a waste basket diagnosis. So perhaps you could take that up and actually comment, how actually is triple-negative breast cancer defined? And what's the most and best established techniques in order to define it in routine clinical practice?
Highly esteemed opponent, thank you for your question. This TNBC is actually, I mean, on the first, on the one hand, it is classified, of course, by the receptor status. So if we have an absence of ER/PR and HER2 receptor, then it is classified as TNBC subtype, but still TNBC has already a lot more subtypes, and to really for example, get a personalized treatment for a patient, you really have to check more than only the receptor status, of course. So what's special also about TNBC, for example, is this high occurrence of BRCA mutations, for example. That also it has very high recurrence rates, also compared to other subtypes of breast cancers. The stem cell population is higher, for example, than in other subtypes, and also the microvascular density, which all makes it like a very poorer prognosis, actually. So it all comes together, and this makes like TNBC this special aggressive subtype.
Thank you very much. And what would be the best way in routine pathology practice in order to define it? Like, what technique do we use? How do we actually detect the absence or the presence of the receptors?
I mean, the easiest way would be like an immunohistochemistry, of course.
True.
So I think this is also in the pathology the first way to go always.
Absolutely. Now, if you're the clinician, you've taken the biopsy, and there is no expression of estrogen, progesterone, and HER2,
Yes.
can you always be very absolutely sure that the breast cancer, which is still in the patient, is definitely triple negative, or are you overinterpreting the results by saying the biopsy is negative?
I would say actually that you cannot really sure of, because it depends on where you have the biopsy taken, and so especially the TNBC is a very heterogeneous cancer, and breast cancer in general, and if you may be unlucky and you just reach a part of the tumor which is like triple negative, but the rest isn't, for example, then of course it's not like a real TNBC diagnosis. So yeah, you would have to check, you would have to check like more places in the tumors, or even check the metastasis. They also sometimes differ from the expression. So
Yep.
yes.
That's what I wanted to hear. Thank you very much. And you already commented that triple negative breast cancer, generally speaking, behaves differently than, let's say, a luminal A type classical breast cancer. Could you outline that for me a little bit more?
So actually, I'm not quite sure how luminal A breast cancer behaves itself, but yeah, what I know about the other subtypes is actually that they are treated a lot better compared to TNBC, because of course you also have these receptor expression which lack in TNBC, and also mostly they have like not such an aggressive growth as TNBC. So the tumor grade is also often lower compared to TNBC at the time point of diagnosis.
Yep.
Also, the age group of women is usually older compared to TNBC. These are mostly pretty young women which gets it, which also makes the relevance even higher to find some treatment.
That's fine.
Yeah.
Thank you.
Okay.
That's concise. That's fine. You already said like the potency of triple negative breast cancer to metastasize is higher, which is absolutely correct and part of the aggressiveness of the disease. Where does breast cancer in general tend to metastasize to?
So in the case of TNBC, it's often the lung, the brain, and also the bones.
Yep.
So these are the main points actually where the metastases tend to form, yeah.
Yeah. And as you said very quickly in that, liver also. If you have a cancer specimen in a patient, which perhaps not has been perfectly staged, right? Mm-hmm. And you have a biopsy and it's an adenocarcinoma and it's a triple negative, what would be an important differential diagnosis in the breast that you always have to think of if a cancer doesn't express the classical markers you would expect to find in breast cancer?
I mean, if the classical markers would not be or could not be analyzed somehow, then yeah, you should take into account some different biopsy methods also maybe, or even do a PCR. Or you could check also for other mutations, because we have a few mutations which are very prone to cancer in general, or even special for breast cancer. So these were...
Thank you. It's great. You do that, and somehow the mutation findings don't quite align. You know, it's not that your cancer, or not your cancer in the patient is not that well is rather well differentiated. Still, it doesn't express any of the regular markers like estrogen, progesterone is all missing. And you've tested for the classical like PIK3CA mutations. They are not present either. What do you have to think of? What could that cancer also be?
Okay, I have to shortly think about this.
It's fine. It's a nasty question, I have to confess.
So, okay, after the after the typical markers are not expressed. I'm afraid I don't know the answer.
Fine. Far for specific, it was not a nice question to ask. It could always be, as we just talked about metastasis, it could be a metastasis to the breast.
Yeah, of course.
Which mimics breast cancer and triple-negative breast cancers.
Yes.
And which is in some really good studies out there where patients were included which actually never suffer from breast cancer at all. But that's like on the fringes.
I see. Yes.
Very concise. Thank you very much. And I would return the word to the prorector. Thank you.
Thank you. The opposition will be continued by Professor Dijkgraaf, Professor of Biochemistry at Maastricht University. Professor Dijkgraaf was member of the assessment committee, and she is the secretary of this degree committee.
Yeah, thank you, Prorector. Dear candidate, first of all, I would like to congratulate you on the completion of your thesis. It was really a joy to read, and of course, I would also like to extend my congratulations to your whole promotion team. But to kick off with my questions about your thesis, I have a somewhat more introductory question. And that's already on page 3. There you introduce cancer, breast cancer. And then I was a little bit disappointed when I read just below figure 1. There you state, despite progressing research, incidence and mortality are still increasing. And then I thought, we are doing so much research, there's proton therapy, immunotherapy. Do you have any reason why is the mortality still increasing?
Highly esteemed opponent, thank you for your question. My guess actually would be that although we have a lot of progress actually in research and also in the breast cancer treatment actually, I think one reason could be also that on the one hand, the people are getting older and older, and therefore also the cancer cases actually increase.
So actually what you're saying is then because we are doing so good in other healthcare parts, this is still increasing, to say it like that.
Yeah, I mean. Yeah, I mean, I think it would be actually a factor that if the people are getting older, somehow that the chance of cancer increase with age, because it's actually also a risk factor.
Yeah.
So I would say this contributes to it, of course.
Yeah.
But yeah, still I also find it like shocking actually, because we think we have so much treatments.
Indeed.
And it's still. Yeah, some types are really hard to treat, actually.
Yeah. Okay, but we can discuss this further after your defense. I would like to go with you to chapter 3. And when I read throughout your whole thesis, actually, I had the impression that PSMA is a little bit like integrin-like the alpha V beta 3 integrin. Concerning the expression, because it's expressed on the vasculature, the tumor vasculature, but also on the tumor cells itself. And then I have a look at, also on page 57, and then figure A, and also the panels D and E. Because if I'm correct, I do see indeed that some of the breast cancer cell lines do express PSMA themselves. Is there an advantage or disadvantage that it's also expressed on the tumor cells?
Yes, in theory, of course, it would be also an advantage if it would be also expressed in the in vivo model. But this was for example, a thing in our model that we could not see the same PSMA expression in the breast cancer cells compared to the in vitro studies, and also in the in vivo model. But yeah, of course, the advantage would be, of course, that you have a dual targeting approach and that you can not only damage the vasculature, which supplies the tumor and is like more of a passive approach, but you could also directly kill the tumor cells, of course, with it.
Yeah, indeed. Yeah. So I fully agree with you that this is really an advantage if it's expressed on the tumor cells itself as well. I also have a question about the isoforms, and this was already addressed by my colleague, Professor Suhrhausen, because I do see a lot of isoforms that are expressed, and mainly on the vasculature of the tumor cells that are induced by the tumor cells. How effectively can the PSMA radioligand target these isoforms? Do you have any idea how effective lutetium PSMA or gallium PSMA can target these isoforms? Is it as effective as to the native PSMA?
That would highly depend on the isoform itself, because some of the isoforms, for example, are located cytosolic, so there would be a problem with having no cell surface receptor for the ligand. That would be one problem. But in general, if the PSMA isoform expresses the transmembrane domain, then in theory the ligand could target it.
Okay, yeah, I fully agree. If it's intracellularly expressed, then you have a problem, because you have to get it into the cell. But how could you set up a test whether the isoforms, if they are expressed on a cell membrane, how could you test whether your radioligand is still binding as effectively to these isoforms? If you are thinking about biochemical assays. Is there a way of maybe mutating some cells with these isoforms, or could you think about some other biochemical techniques to test this?
I mean, in theory, you could overexpress the special isoform in, for example, in cells or in some bacteria or in some organism, and then you could like do uptake studies to check if there are any differences in the binding behavior of the same ligand with different isoforms. So that would be a possibility to check the binding, maybe the differences.
Thank you very much. I think for the sake of time, I will give the word back to the prorector. Thank you.
Thank you. The opposition will be continued by Professor Eble, Professor Radio Oncology at Aachen University. Thank you also for joining this ceremony.
Thanks. Dear Mrs. Candidate, first of all, it was several times said, but it's very important. Congratulations also from me for this excellent scientific work. When I started being a radiation therapist to read the different papers, first of all, I need some time to get really in detail, but nevertheless, more and more I was very fascinated from the aspect to differentiate between a really cell-associated treatment or a treatment being indirectly against the components of surrounding the tumor cells. And this is also the point of my question. So when we go to in chapter 3, the figure 5 and 6, we could see that opposite to the uptake in prostate cancer, which you all know, and which is a direct treatment to the prostate cancer cells, we could see that there's also an uptake for the breast cancer cell lines. It's really less. And we could see two other things, that there is a combined effect with the endothelial cells or the combined cells, and also we could see that the apoptosis is, of course, then really higher, significantly higher if there is a combination of co-cultures with the endothelial cells. So the first question is, what is really now the target? It's the cancer cell, or it's the components around the cancer cells? Could we see this with when I look for the apoptosis? It's a completely increased apoptosis, but it's on the tumor cells. You mentioned that, or it's on the endothelial cells, which lead then to the death of the tumor cells.
So, did I understand that right? That you mean that what the tumor cell is dependent on to express this PSMA? Or?
Yeah.
Yes. This also has a lot to do with the surrounding, especially with these growth factors which are secreted, which stimulate them and promote this pro-angiogenic behavior in the endothelial cells. And for example, what I found very interesting is that the TNBC cell lines induce a lot more to the prostatacarcinoma cell line. And this I read in a study, they checked this behavior also, and they actually saw that this has also to do with the androgen receptor expression. So it's a very highly regulated and complex process, and it's dependent on a lot of things, this PSMA expression.
So it's finally an anti-angiogenic effect, or it's a direct tumor effect using the PSMA?
So actually, if androgen is decreased, then PSMA is actually increased. So this has a direct effect on each other.
Mm-hmm. You then discussed in the conclusion that fractionation may be an important point in increasing the efficacy of this treatment. What was the rationale for thinking about the fractionation?
I mean, usually why would you use fractionated therapy is, of course, that you could deliver like a dose of treatment in shorter time periods. So in theory, it would be more efficient than if you only give one dose, but of course the fractions have also to be high enough. So this is why I thought that in my treatment the single dose worked better, because usually the advantage of the fraction is that you continuously deliver smaller doses of activity to the tumor than if you only do one shot.
You mentioned in the chapter 4 the term reoxygenation, mm-hmm. which is a very important point in irradiation of tumors. What's mentioned with that, and also in correlation with your results?
I mean, I think oxygenation is very important if you have, for example, tumors which are resistant to radiation. I mean, there are tumors that are more receptive to radiation and some are not. And to overcome this often hypoxia-induced resistance, you can oxygenate the tumor tissue and therefore make the tumor more sensitive to the following radiation treatment.
It's correct that the radiation sensitivity is really low with having hypoxia in the tissue. But it's then the correct way to do a fractionation if you mentioned it could be an anti-angiogenic treatment? You also saw in your in vivo experiments that there are a lot of necrosis in the tumor,
yes.
And therefore also I think a lot of rims with hypoxic cells around the necrotic areas. It's then also doing an anti-angiogenic treatment the correct way to do a fractionation, to lead reoxygenation?
That's a hard question.
It's a hard question, yeah. It's not your point because you're not a clinician, but I think it's an important question thinking about on doing the specific
If you ask
treatment.
like that, then I would probably say no. But
it's a good answer.
Yeah, I mean, I think if I think about it, it could be also it could have to do something with that. If we target the blood vessels, then we somehow induce hypoxia also in the tumor, because we have less blood supply. And maybe this is why fractions are maybe not so suitable in this case than single doses.
Thank you very much. It's a very specific point. I have no other questions concerning that.
Thank you. The opposition will be continued by Professor De Ruijser, Professor Radio-Oncology at the Maastricht Institute for Radiotherapy here in Maastricht.
Thank you very much. Also, I want to congratulate you and your whole team and also your family for this big endeavor, because a PhD is always very cumbersome for many people. But I'm here to ask some questions. And actually, you mentioned all the time, actually, in all your answers and also in your layman's talk, that the single dose is actually better than the fractionated dose. But if I look at least to the survival curve, I would argue that these are really superimposable, whether you give fractionation or not, because actually it's only based on a small number of mice. And it's only if you have one mouse more dying at an earlier time point, only one, then it will be exactly the same. And if you would do, it's not really possible because of the small numbers. A classical statistical test, I would argue that there's really no difference at all if you look at the curves. Do you agree or disagree with me?
Highly esteemed opponent, thank you for your question. I would actually partly agree, because yes, on the one hand, you are right that I actually used a small mouse number, and also the differences in the survival curves were not statistically significant. But I mean, still, I think there is still a trend, because we had this significant differences regarding the tumor volume, and also the time which, for example, the mice needed to reach 200% of the tumor volume. So therefore, I would say that in our case, for the activities we used, the single dose worked better. But also this, of course, if you use higher fractions, then maybe the fractionated then would still work better, so that we would have to try.
If you look at the volume, actually it's only one point where there's a difference, and that's it. So I would argue, again, probably there's no difference at all. You should redo the experiment, and I know that it's easier said than done, I know that. But on the other hand, I still want to make the point that maybe you overinterpret the results on the basis of, a small number, and trends in statistics, that's a chapter on its own, of course. What can be shown as a trend, or do you want to show as a trend? But coming back to the issue about the survival again, apart from where we partly disagree then about fractionation or no fractionation, I see that all the mice die. So I don't see any cure. Why is this?
Yes, this is true. This treatment was not curative. I think this has several reasons. On the one hand, for example, this beta-minus radiation of the 177 lutetium is not as aggressive, for example, as alpha radiation. So if you would, for example, use alpha radiation instead, then you could have better chances of a higher effect of the treatment. Yes, this has also something to do that the beta-minus radiation, for example, induces mostly single-strand breaks and no double-strand breaks in the DNA, which makes it more reparable in theory for the cell. So this would be maybe one option to come closer to the goal of curing.
I'm very happy with your answer, because actually I've written down, why wouldn't you go to alpha radiation? Because hypoxia throughout your different chapters is, of course, a big problem. And as we know, in the human situation, I don't know any tumor which has not some hypoxic cells. So again, my question is then, I agree with you, alpha radiation, but would you continue to do studies with lutetium in the framework of PSMA treatment, or would you say, well, you know, you're quite young. So that means that I wouldn't go for something which fails in even preclinical models. I would directly go for something else. Because it's a nice thesis, huh? But wouldn't you go for something more ambitious?
You mean in case of radionuclides or in general?
In case of radionuclides, yes.
So in case of radionuclides, I would probably, I mean, if possible, and also if possible to work with it, because not every lab is, of course, suitable for alpha radiation. But I would find it very interesting, actually, to see if the alpha, for example, there is already 225 actinium PSMA, which is used in studies. So this would probably be very interesting to exchange the beta-minus radiation with the alpha radiation.
And would you go for the same mouse model, or would you go for another mouse model if you want to do preclinical experiments with the alpha emitter? And I want to come to the possibility of the interaction between the immune system and the effect of your treatment.
Sorry, I forgot the first part. Can you please repeat?
Well, actually, we treat in clinical practice triple-negative breast cancer or some subtypes with immune treatment. And I want to come to, and sometimes, at least preclinically and also in clinical studies, which we also did, that is, there's a quite interesting interaction between, let's say, classical forms of anti-cancer treatment, including radiation, and immune enhancement. It's very complex. We don't understand even half of it, I'm convinced. But still, would you go for a kind of preclinical experiment where you can address this question?
Yes, I think that actually the tumor microenvironment offers a lot of therapeutic targets in theory. But if you, for example, could combine like several approaches, like, for example, the targeting of the tumor vessels with a PSMA ligand, for example, in combination with maybe a checkpoint inhibition or some other therapies, then you could also really increase the efficacy of the treatment, of course, because you also had like a more targeting approach, like a dual targeting approach. And yeah, I think that would really increase also the efficacy of this treatment.
Thank you very much. This was really very efficacious. Thank you. I give the word to Mr. Prorector.
Amelie Heesch, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return to this room.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you grow. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Amelie Heesch, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Motagui is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer to you, Amelie Heesch, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. So, now photos, please. So. Oh no, this doesn't work. I'm doing it wrong. So like this. So this is then you just keep it like this. That's for you.
Thank you.
Congratulations.
Thank you very much. Please be seated.
Now it's time to say some words to you, some personal words, of course. Maybe just briefly all of you know you very well, but maybe a brief step towards your CV, which is something that's important also since we are in Maastricht. So you were born in the Netherlands, in fact, in Amsterdam, and also your name has some Dutch background with a double E. So you should be somehow familiar in Maastricht, and great day you did today. So, Dr. Amelie Heesch, which I may name you now, congratulations. So you came to our department in 2020. It was just before the COVID time, actually. Actually, when you applied for the position, it was in 2019, and you started in January 2020. And so you had really not the best time at the beginning with all the COVID stuff, and also in the lab it was difficult, but not only due to COVID, but also due to the task you got. So it looked so easy with all the in vitro models and so on, and this was not easy at all. So it took a lot of time and a lot of interaction with different co-workers, with different other departments in the hospital to really get this running. And finally, you were able to have this nice model, and also this in vitro results showed some very nice aspects, and as you presented today. And what was important in that, I think that's also a very good strain of you, that you still kept being motivated. You were, to some extent, of course, frustrated, that's normal, but you still went on and said, okay, I really want to do it. And that's something you should keep for yourself also, that you try not to give up and just go on and try to move into that direction. I think that was an important aspect, and also to learn from you that you are such a direction-minded person going in that direction. During your PhD thesis, you had a lot of topics to learn. I mean, we saw today, it's not only the in vitro, it's also looking into the how to use a microscope, how to interpret data, and well, using microscope is easy, but how to interpret the images, and also to go into the different kind of what we heard from the pathologists. I mean, they are usually sitting the whole day after the microscope. So, and you try to imitate it to some extent, and you did a great job in that. And important aspect, but not only that, also the in vivo work part of your thesis, having the right animal model, all these kind of things. There you really used the knowledge of the department, but you also brought in your own knowledge, also what you achieved already during your master thesis back in Kaiserslautern, I think you did your master thesis. So I think with this variety of skills you have, so it should be easy to find a next job and also next step. So it seemed to be not the easiest approach in nowadays because you're still trying to find something, but I'm sure you will find an adequate position at some point, and then hopefully the people that you will be working with will enjoy working with you like we all did enjoy working with you, because you're very social and very easygoing person to work with. And I think that can be said also by your co-workers. And so stay like this. That would be great. And yeah, just that leaves me to the point for your future. I wish you all the best, and hopefully also together with your family, then you can decide whether you really want to stay in Aachen. You're living now in the most beautiful part of Aachen, in Frankenberger Viertel, yes. Which is, I can understand, difficult to move away, but maybe at some point you need to decide that to go to find a different position. But yeah, you will do the right way. All the best for you.
Thank you very much.
Dear Dr. Heesch, also on behalf of the Maastricht University and the Board of Deans of Maastricht, it's a pleasure for me to congratulate you with the degree you just acquired. As already mentioned by your supervisor, the university also wish you all the best in your future career and a lot of success in your future research. Hereby, I will close this academic ceremony. Before we are going to congratulate the new doctor, we will take a picture here in the aula in front of the screen so that Professor Gonfin is also on the picture, and afterwards the on-site members of the degree committee will take a picture with the new doctor on the stairs in the central hall. Hereby, I close this academic ceremony.
Come in the front.